Good day, everyone. I'm Lavanya Raajaraam, a PhD scholar working in the Indian Institute of Technology, IIT Madras, India, and I'm guided by my research supervisor, Dr. Karthik Raman. Today, I'm here to present a flash talk on our poster, which is about the algorithm Co-FSEOF, which helps us to study co-production of different metabolites. So we know that a lot of commercial products can be produced using bio-based systems, but still the adoption in industry level is limited because of the low feasibility and yield. So, in order to improve the economic feasibility of a given process, we can couple the production of two or more products. For example, it has been shown that riboflavin and butanol can be co-produced, where one is a high-value product and the other is a low-value product. Or we can also choose the product such that one is accumulated intracellularly, like PHB, and the other is secreted outside, like succinate. So, in the end, the downstream processing costs also go down. And in some cases, like uridine and acetone, it is seen that co-production is more favored when compared to the production of single metabolites because it ensures better cofactor balance and better carbon usage. So, apart from the economic feasibility and better cofactor balances, it just helps us exploit the system in a better fashion and utilize the resources in a better fashion. And in cases like biofuels and fatty acids, we need to co-produce a cocktail of metabolites rather than a single metabolite. So, in those cases, also co-production is very useful. So our objective was to develop an algorithm to study the co-production of metabolites and study its performance for multiple metabolites and higher order interventions. So higher order interventions are two or more amplification or knockout of genes. So here we have also studied the application of the algorithm to commercially valuable products like fatty acids and biofuels. The work done, we have studied the co-production for all the secretory metabolites in E. coli and SVCA under both anaerobic and anaerobic conditions, and we have obtained higher order intervention strategies, especially for the co-production of isobutanol and succinate in SVCA. So the workflow goes like this. We apply FSOF for a single product and find the common targets for two or more products, and we validate them using flux variability analysis and choose the intervention strategies with the best score. So I'll explain the workflow in a detailed fashion. So we have the genome scale metabolic models of the organism, which is represented as a stoichiometric matrix of reactions and metabolites. We perform flux balance analysis on this organism. The organism can exist in multiple states, two out of which are maximum biomass production and maximum product production. So we enforce the organism to move from this state to this state, and as it how the flux through the other reactions in the system changes. The reactions that undergo increase in flux are potential amplification targets, and those that undergo decrease in flux are potential knockout targets. And we obtain such targets, and we can separately optimize product A and B. But if we apply the intervention to the common targets, we should be able to co-optimize both product A and B. And this is what we see too. Here are some commercially valuable metabolites that we have been able to co-produce. You can see that the wild type flux is pretty low, but when we amplify or knock out certain reactions, we are able to see an increase in the mutant product flux, but there is a decrease in biomass flux too. So the score is given by increase in the ratio of increase in product flux to ratio to the decrease in biomass flux. We have the scores for separate products and also for the whole process as a whole, which is the average of these two scores. And we can see that a lot of commercially valuable products and biofuel candidates are present here. We have also simulated this for anaerobic conditions for time constraint. I'm skipping that. And we have also shown that we can obtain higher order intervention strategies where we have combinations of knockouts and amplifications, and also multiple knockouts and amplifications. Here I have ordered them according to the number given here. So for example, you can see that the red ones are knockout targets and the pink and blue ones are the amplification targets. And this is actually a snapshot of the glycolysis and TCA cycle in SVCA. Here you can see the products succinate and isobutanol. You can see that a lot of targets are actually placed in the production pathway itself, as one would expect. Here you can see that, for example, in number one, you can see the knockout of these two reactions and the amplification of this reaction is able to give us good increase in both succinate and isobutanol fluxes. We see that some reactions are also present in some auxiliary pathways that belong to nucleotide metabolism and shikimate pathway, etc. So this shows that the algorithm is able to perform well in identifying non-intuitive targets. So to summarize, we have co-produced a lot of commercially valuable metabolites in both E. coli and SRVCA. And we see that anaerobic conditions support the co-production of more combinations of metabolites. And if we identify higher order intervention strategies, we can expand the number of combination of metabolites that can be co-produced and also improve the flux that we can achieve. I'd like to thank everyone for your patience and for listening to me. And I'd like to thank my guide, my institute, and my lab members for all the support they have given me. Thank you for the opportunity, and I'd be happy to take any questions that you might have. Thank you.